Researchers want to create fresh therapies for cystic fibrosis and other lung conditions using these RNA delivery particles. An innovative sort of nanoparticle that can be injected into the lungs and transport messenger RNA encoding beneficial proteins has been developed by scientists at MIT and the University of Massachusetts Medical School. These particles may eventually provide an inhalable treatment for cystic fibrosis and other lung conditions, the researchers believe. This is the first demonstration of highly efficient delivery of RNA to the lungs in mice. We are hopeful that it can be used to treat or repair a range of genetic diseases, including cystic fibrosis, says Daniel Anderson, a professor in MIT's Department of Chemical Engineering and a member of MIT's Koch Institute for Integrative Cancer Research and Institute for Medical Engineering and Science. In a study involving mice, Anderson and his associates delivered mRNA encoding the tools required for CRISPR, Cas9 gene editing using the particles. This might make it possible to create therapeutic nanoparticles that can cut out and replace disease-causing genes. The therapeutic use of messenger RNA in the treatment of many diseases brought on by defective genes has considerable potential. Its deployment has so far been hampered by the challenge of getting it to the proper portion of the body without experiencing off-target consequences. There are now multiple clinical trials examining potential mRNA therapies for liver disorders since injected nanoparticles frequently build up in the liver. Direct injections into muscle tissue of RNA-based COVID-19 vaccines have also shown to be efficacious. Numerous times, mRNA is contained within a lipid nanoparticle, a fatty sphere that prevents mRNA from being prematurely degraded and facilitates its entry into target cells. With the goal of better transfecting the epithelial cells that make up the majority of the lining of the lungs, Anderson's lab set out to create particles a number of years ago. In 2019, his lab developed nanoparticles that might infuse lung cells with mRNA for a bioluminescent protein. These particles were easier to aerosolize for inhalation into the lungs because they were formed of polymers rather than lipids. To improve those particles' potency and maximize their utility, more work must be done on them. The goal of the researchers' latest investigation was to create lipid nanoparticles that might specifically target the lungs. The molecules that make up the particles have a positively charged head group and a lengthy lipid tail. The head group's positive charge aids the particle's interactions with mRNA's negative charge as well as mRNA's escape from the cellular structures that enclose the particles after they enter cells. In the meantime, the lipid tail shape facilitates the particle's passage through the cell membrane. For the lipid tails, the researchers proposed 10 distinct chemical configurations, along with 72 possible head groups. The scientists were able to find the various combinations of these structures that were most likely to reach the lungs by screening them in mice with various combinations. Further experiments in mice demonstrated that the particles may be used to transmit mRNA encoding CRISPR, Cas9 components intended to remove a stop signal that was genetically programmed into the lung cells of the animals. A gene that produces fluorescent protein activates when that stop signal is deleted. The percentage of cells that were effective in expressing the mRNA can be calculated by the researchers by measuring this fluorescent signal. The researchers discovered that after one dose of mRNA, around 40% of lung epithelial cells were transfected. The level increased to more than 50% after 2 doses and to 60% after 3 treatments. 2 epithelial cell types called club cells and ciliated cells, each of which was transfected at roughly 15%, are the most crucial targets for treating lung illness. This means that the cells we were able to edit are really the cells of interest for lung disease, Lee says. This lipid can enable us to deliver mRNA to the lung much more efficiently than any other delivery system that has been reported so far. The risk of inflammation is decreased by the new particles' fast breakdown, which enables them to be removed from the lung within a few days. If repeated dosages are required, the particles could likewise be given to the same patient more than once. They have an advantage over a different method of mRNA delivery that makes use of innocuous adenoviruses that have been modified. Although those viruses are highly good at delivering RNA, they can't be used repeatedly since they make the host's immune system react. This achievement paves the way for promising therapeutic lung gene delivery applications for various lung diseases, says Dan Peer, director of the Laboratory of Precision Nanomedicine at Tel Aviv University, who was not involved in the study. This platform holds several advantages compared to conventional vaccines and therapies, including that it's cell-free, enables rapid manufacturing, and has high versatility and a favorable safety profile. The researchers employed an approach known as intratracheal instillation, which is frequently used as a way to imitate administration of medication to the lungs, to administer the particles in this investigation. They are currently striving to increase the stability of their nanoparticles so that they can be nebulized and aerosolized for inhalation. The particles will also be tested in a mouse model of cystic fibrosis to see if they can carry mRNA that could fix the genetic mutation discovered in the gene that causes the condition. They also intend to create mRNA vaccines that might be administered directly to the lungs, as well as therapies for additional lung conditions such idiopathic pulmonary fibrosis. The American Cancer Society, the National Institutes of Health, Translate Bio, the Leslie Dan Faculty of Pharmacy Startup Fund, a Prime Postdoctoral Fellowship from the University of Toronto, the Cystic Fibrosis Foundation, and the Prime Postdoctoral Fellowship provided funding for the study.